In the chloroplast of a plant cell, there is a thylakoid. In the thylakoid, there is a structure of proteins, which includes photosystem 1 and photosystem 2. In photosystem 2, there are electrons. Light energy comes in and charges these electrons. These electrons move through a carrier protein to another protein. Now, the electrons moved by the carrier protein have to be replaced in photosystem 2. These electrons are replaced by electrons taken away from water molecules. Once these electrons are replaced with electrons taken away from water molecules, this leaves us with oxygen gas as a byproduct. Now, these charged electrons are used to pump hydrogen plus ions from the stroma, which is outside the thylakoid, into the thylakoid. This then creates a concentration gradient. This concentration gradient powers a protein that has the ability of ATP synthase. This protein takes in ADP then ATP comes out. Now, the electrons used to power the pumps to transport hydrogen are transported into photosystem 1. Now, light energy comes in and charges the electrons. Now, the charged electrons are transported with another protein onto another protein. Now, these charged electrons are used to reduce NADP plus to NADPH. The NADPH now goes into the stroma. All of these reactions are called the light-dependent reactions. Now, in the stroma, there are many ATP and NADPH. ATP and NADPH produced in photosystem 1 and photosystem 2 are used to fuel the Calvin cycle. In the beginning of the Calvin cycle, RUBP is joined by carbon dioxide. This results of a 6 carbon molecule. They then split into 2 3-carbon molecules. The first step is called carbon fixation. Now, for the second step, the 6 carbon molecules go through a series of reduction. ATP comes in, then ADP comes out. NADPH comes in, and NADP plus comes out. After the reduction, G3P is created. In the final step, RUBP is regenerated. After 3 cycles of the Calvin cycle, 6 molecules of G3P have been created. 5 of them are used to regenerate the RUBP. ATP goes into the 5 molecules of G3P, then ADP comes out. After this, 3 molecules of RUBP have been regenerated to keep the cycle moving. The remaining G3P is joined by another G3P, which also remain from the Calvin cycle. They are then used to produce glucose. Then the phosphate can be removed. Then fructose would be added to create sucrose. The glucose phosphate is also the starting molecule for the synthesis of starch and cellulose.